We're now going to deal with the most important process yet when it comes to DNA. That is the process, as it states here, of protein synthesis. All that means is that we need to make protein. So please understand the big picture here. We've spent a lot of time throughout the last few weeks talking about different ways that genes are transferred from one generation to the next. We've talked about different versions of those genes. Some were completely dominant, others were incompletely dominant, others were co-dominant, and they're sex-linked and multiple alleles, etc. All of that comes down to one simple fact. All of those traits, all they really mean is that they are a code, and it's a code for your body to know how to make the correct proteins. That's what your traits are. Your traits are the result of the DNA within each one of your cells, and that DNA simply is a four-letter code, A's, T's, C's, and G's, that explain to each one of your cells how to make a correct protein. You are the result of your body making proteins, making them correctly, making them in the right quantities, making them at the right times throughout your life, but you are simply the result of making proteins. And so you can't really overstate the importance of this process. What you're looking at in this particular picture here is supposed to represent one gene. In other words, you're taking one chromosome and then one segment of DNA within that chromosome, and that's going to represent a code that is a gene. And that gene is there to provide the code for making a specific protein. What you are looking at here also is that not every portion of your DNA represents a gene. For instance, right here and right here, both before and after the gene in question that we're looking at, those are not protein coding portions of DNA. All of this process of protein synthesis involves chemical reactions, and you know that chemical reactions rely on enzymes. So the enzyme we're going to focus on right now is the enzyme RNA polymerase. It's got to start this whole process of protein synthesis, and it has to know exactly where to start and also where to stop. So the RNA polymerase enzyme is going to work its way through the DNA, and eventually it's going to come upon a certain portion of DNA that it recognizes as what is called the promoter. It is the promoter region of the gene. And so it knows when it sees a certain sequence of letters, it knows that what comes next is in fact the gene, where it will start to do its job of reading the gene and performing the whole process, beginning process of protein synthesis, but eventually also it needs to know when to quit. And so this segment of DNA right after the gene is known as the terminator region. So it tells the RNA polymerase enzyme, oh hey, it's time to stop. So this enzyme knows where to start and knows where to stop based on the promoter and terminator regions. In between there is the gene. In other words, is the code for making the protein that is supposed to be made. That whole process of making a protein is going to be broken down into two steps. The first of those steps is called transcription, and it's going to take place with the DNA, of course, in the nucleus, because that's where our chromosomes are housed. What you're looking at here is supposed to be the DNA from the previous picture. So here is the top portion of DNA, and all I have written are the letters to represent the bases that compose these nucleotides. So here's the top portion, and down here the bottom portion. So if we go back to the previous slide, here's the top portion, here's the bottom portion. The RNA polymerase enzyme, after it found the promoter, one of the first things that it did was to separate the two DNA strands, the two halves of the strand, into a top and a bottom portion. In our case right here, this top portion, the series of letters above is known as the sense strand. And it's those letters that, frankly, are not important to us. They are not going to be used. It's the bottom letters down here, or the antisense strand, that are the code. This is the code for making the protein that this gene is supposed to make. The RNA polymerase enzyme comes and it starts to read the antisense portion of the DNA. And so if we start over here on the lower left, it reads C, and so its job, the chemical reaction it performs, is to add free-floating RNA nucleotides. I stress the RNA because what's written here in blue, the G in this case, is not a DNA nucleotide, it's an RNA nucleotide being added to the DNA. So the C is DNA, the G is RNA, and they are being linked together by the RNA polymerase enzyme. The enzyme then just reads a T, so it adds an A. It reads a G, so it adds a C. And it does the same thing again and again. Now let's focus specifically on this A right here. You know that A's always go with T's, but what you've also learned is that RNA has no T. So instead, the letter in blue here is going to be a U. A's go with U's. So this is uracil. Anytime you have a blue letter, it's either going to be G or A or C or U. Anytime you think, oh, I should add a T, no, it's not a T. It's a U for uracil. And so the RNA polymerase enzyme is going to work its way the whole way down the antisense strand, adding RNA nucleotides, free-floating RNA nucleotides the whole way through until it comes to the terminator region. And then this G is the last one it's going to add, and then it is told by the terminator region it's time to stop. What you have just created there in the blue is called pre-mRNA, or pre-messenger RNA. Now, why is it pre? Well, it's pre because sometimes there's some problems with the letters in the antisense strand. You can see that I have a block of red here and a block of red here. We have two different types of segments of DNA here. We have what is in the red, which are called introns, and we have what is not in the red, which are called exons. The introns, I-N-T, introns, are named that because they are intervening. They're in the way. In other words, they're not really supposed to be there, and there's different theories as to why they still are, but for our purposes today, in making the protein that your body is supposed to make, this GG combination in this case and this TT combination in this case are intervening. They are not supposed to be there. They are in the way. They are introns. All these letters that are not in red are exons. They are supposed to be expressed in the form of our protein. So these are what we want. These are what we don't want. And so we have to have a process of getting rid of this AA and rid of this CC because they're just not supposed to be there. That process, I'm going to skip over this picture here for a second. That process is done by, and don't laugh, things called SNRPs. Small nuclear ribonucleoproteins, you can see why we have to have a nickname for them, are called SNRPs. And those SNRPs bundle together, here you can see a group of 4 of them, into a structure called a spliceosome. The spliceosome takes the exon, there's an exon in red, there's another exon in red, and this pinkish part in between is the intron, and you can see what's starting to happen. This red part is going to be pulled in this direction. This red part is going to be pulled in this direction. And these two red portions are going to be put together, and this pink portion is going to be cut out. So the red portions are spliced together, thus the name spliceosome. And the pink portion, the intron, the intervening portion of it, is cut out, as you can see here, and is gotten rid of. And so in the end, transcription will finish, and all of the code, the mRNA code, is what is supposed to be there. And at that point, the mRNA is no longer pre-mRNA, it is functional mRNA. And as you know, the M stands for messenger. That messenger here will detach itself from the DNA, and it will deliver the message. In other words, the code for making these proteins is, of course, in the nucleus, which is where transcription is taking place. But where do proteins actually get made? They get made in a ribosome. So a message has to be delivered. This mRNA, seen here, will deliver that message. It will detach itself from the DNA. It will leave the nucleus via those nuclear pores that you learned about. And it will go to a ribosome so that the second half of protein synthesis can take place.